Assalamu alaikum. Today we'll be covering the Golgi apparatus and vesicular transport as lecture number 2. So last time we talked about endoplasmic reticulum. Now we'll talk about the Golgi apparatus. It's the step that comes after ER. So these are the processes that take place at the Golgi apparatus. Okay. So what happens is that some proteins are processed in the ER. They get glycosylated, for example. They have attachment of lipid molecules as well. That continues in the Golgi apparatus. Also, in the Golgi, you have sort of like sorting of proteins. So proteins that would go to peroxisomes would be allocated in certain vesicles. Lysosomal proteins, same thing, plasma proteins, secretory proteins, and so on. And then you have synthesis of glycolipids and sphingomyelin that takes place at the Golgi apparatus. So this is how the Golgi looks like, okay? So what you have in here is basically the Golgi looks like stacks of sacs, cisternae, okay? So, and there are 4 regions, and these are the cis, because it's the one closer to the endoplasmic reticulum. You have the medial, the trans, and the trans-Golgi network. That's where vesicles start to pop up of the trans-Golgi going into different places. Again, plasma membrane, secretion, endosomes, lysosomes, and so on. Okay, so the way proteins move is that the Golgi sort of like progresses. It matures, or it changes location. So you have continual regeneration of the Golgi apparatus. So vesicles come in from the ER, and you have movement of these sacs until you get to popping up the vesicles out of the Golgi, and some of them go back. Okay, so some of them go back and they fuse with the previous sac. Okay, or that they fuse with the previous sac. So now what happens in the Golgi apparatus is that you have continual modification of sugars that are associated with the with proteins, sugars that were added in the ER. Okay, so you have a number of proteins, including mannose. You have further modification of this mannose, and addition of more sugars. One of them is fucose. And notice that the very last one is sialic acid. I think we covered sialic acid in the biochemistry course. Okay. Now, what also happens in the Golgi is all linked glycosylation. So we said that there are 2 types of glycosylation processes that take place. One of them is N-linked glycosylation, which starts in the endoplasmic reticulum. That's where you have sugars added using the lipid carrier dolicol to asparagine. So that's N-linked glycosylation, and that modification continues in the Golgi apparatus. Now, the second type of glycosylation is known as O-linked glycosylation. That's where sugars are added to the hydroxyl group of serine and threonine amino acids. Okay, so so what you have in the Golgi apparatus is this, as well as you have O-linked glycosylation of Proteoglycans. The large molecules that are part of the extracellular matrix, where you have repetitive disaccharides, the disaccharides that are sulfated, they are carboxylated. Overall, they are negatively charged molecules. Okay. Now, what happens in the Golgi apparatus is that you have the formation of sphingomyelin from ceramide. Remember that ceramide is synthesized in the endoplasmic reticulum. It's taken to the Golgi, and in the Golgi it is converted into sphingomyelin. Or it can be converted to glycolipids by the addition of sugar molecules, specifically glucose or galactose. Now, what happens then is protein sorting. Now, something interesting about the Golgi is that Golgi proteins are associated with the membrane, okay, rather than being soluble. Now, the soluble proteins are the proteins that are enclosed in vesicles, and then they pop out of the Golgi, where they fuse with the plasma membrane, with recycling endosomes, late endosomes, and so on. Okay, so what happens then is sorting of proteins in different ways. So what happens is you have this continuous unregulated secretion. So vesicles pop out of the Golgi and they move towards the plasma membrane directly, where they release their contents. Or you have transfer of vesicles to recycling endosomes, and from recycling endosomes you have the release or secretion of these proteins once vesicles fuse with the plasma membrane. So this is continuous unregulated secretion. What you have then as well is the formation of vesicles, including and these vesicles are not mature. They are immature vesicles that undergo maturation by exchanging proteins on the surfaces of these vesicles. Okay, and the other thing is that you have vesicles that are generated from the trans-Golgi network, whereby they are directed to late endosomes, and from late endosomes to lysosomes. So we're talking about lysosomal proteins, or lysosomal content. Now remember that what you have here in these vesicles, you have the membrane, vesicular membrane protein, as well as the lumenal proteins as well. Okay. Now, remember that in specialized polarized cells, like intestinal cells, where you have an apical part or membrane, and you have the basolateral membrane, you have specified vesicles that are directed either to the apical side or to the basolateral side. And this targeting of vesicles depends on the proteins and signals on proteins. So proteins that are targeted to the apical side of the cell, to the apical membrane, they are modified by the addition of GPI sugars, or sugars associated with glycosylphosphatidylinositol. Now, whereas basolateral proteins have a special amino acid sequence, I'm not gonna specify what the sequence is. It's a certain sequence of amino acids. Okay. Now, in terms of lysosomal proteins, what you have in here is it's dependent on mannose, the sugar that is added first in the ER, then it's modified. You have modification taking place in the Golgi, and what happens is phosphorylation of the mannose, one of the mannose sugars. So what you have here is the formation of a mannose 6-phosphate. Now, the enzyme that is responsible for the addition of this processing depends on a signal patch, which is basically a three-dimensional structure that is part of the protein itself. Okay, so it's not really an amino acid sequence. It's rather three-dimensional structure, a signal patch. Now, what happens then is that you have this protein that is that contains the mannose 6-phosphate. Now you have a receptor in the Golgi, and this receptor is a mannose 6-phosphate receptor. It binds to the protein that has the mannose 6-phosphate, and then it gets packaged in vesicles, and they are directed to late endosomes, and then later on to lysosomes. Okay, so the idea here is, how do vesicles form and how are they transported? Now the thing is, you have the membrane proteins and the luminal proteins. They can bind to receptors in the Golgi, and then the vesicle pop out of the Golgi. Now these vesicles that pop out, they get coated with certain proteins, okay? And these coating proteins can interact with the cytoskeletal filaments, okay? Like actin and microtubules, for example. And then these vesicles are transported along the filaments until they hit the plasma membrane. They fuse with the plasma membrane, releasing their content. Now the idea is, what are these vesicles? Basically, there are different types of vesicles. Depending on where these vesicles are heading, these vesicles determine where these proteins are generated from and where they go. So you have what is known as COP2-coated vesicles. So it's special proteins or specialized proteins. These vesicles that are coated with COP2 proteins, they carry the vesicles from the ER to the Golgi. On the other hand, vesicles that are coated with COP1 proteins, they go backward. So they go from the trans-Golgi to the cis-Golgi, from the cis-Golgi to the intermediate compartment, and from the intermediate compartment back to the endoplasmic reticulum. Now you have a third coating in here, which is a protein known as Clathrin, and Clathrin-coated vesicles are targeted from the trans-Golgi to the plasma membrane, or the opposite. So what you have in here is that vesicles are formed from the plasma membrane. They are coated with Clathrin, and then they fuse with lysosomes, with endosomes, and from endosomes to the trans-Golgi. Or these vesicles can fuse with the lysosomes as well. So there are three types of coating molecules, and these molecules or proteins, and these proteins determine the direction of movement of these vesicles: COP1, COP2, and Clathrin. Now, what's important is, how do vesicles know that they have to fuse with this membrane specifically? Now, this is determined by three proteins. What you have in here is, you have two proteins, and they are known as SNARE. Okay. Now, these SNARE proteins, there are proteins that are present on the surface of vesicles, and SNARE that are localized in the target membrane. Now what happens, that's two. The third one is a class of proteins known as RAB proteins, and there are some 20 different RAB proteins, and these RAB proteins determine how these vesicles must be targeted, or to which region they should go to and fuse to. Okay, so basically is this: you have this interaction between a RAB protein with a tethering factor on the target membrane. So this interaction allows for the interaction or tethering, tethering of the snare proteins, the V-SNARE and the T-SNARE. Now they are attached to each other. What happens then is that you have removal of the coat, the Clathrin coat, for example, and that allows for the vesicle to get closer to the plasma membrane, and then what happens is you have fusion of the vesicle with the plasma membrane, so it becomes part of the plasma membrane and the content is released outside of the cell. Okay, then you have removal of the snare proteins from each other. Now, just to connect things to diseases, there is a condition known as Griscelli syndrome, and in this syndrome, it's caused by mutations in the proteins that regulate vesicle fusion with membranes, like the myosin 5A, one of the RAB proteins, and so on. So the idea here, what happens in these individuals is that their hair looks gray. The reason is that there is no good equal distribution of melanosomes that contain the pigments. Now, what happens is that these melanosomes are formed in melanocytes, and these melanocytes have these extensions, and they fuse with the keratinocytes, releasing the vesicles into keratinocytes, and keratinocytes have the color, then the pigments. That's what gives us hair color. Okay, now what happens is that when you have mutations in these vesicular proteins, that would result in unequal distribution of melanosomes, of the pigments in keratinocytes. So you have them looking like this, for example. You have sort of like accumulation of these pigments. Now, so let's talk about lysosomes now. Now, lysosomes are membrane enclosed organelles. They contain a lot of enzymes, such as proteases, galactosidases, and so on. So what they do is that they degrade material. Okay, so they are the waste location of cells. Now, so it contains a lot of enzymes, and these enzymes, by the way, are active at acidic pH. So the environment in the lysosome is acidic, and the reason is that to allow for example, protein denaturation. So at very low pH, what you have in here is proteins become unstable and they get denatured. Once they get denatured, it becomes easier for proteases and other enzymes to act on these molecules. Okay, and the pH inside lysosomes is about 5. The pH inside endosomes, by the way, is about 6. Now, this low pH is maintained by a proton pump that is ATP dependent. So this pump allows for the transport of protons from the cytosol into lysosomes, maintaining an acidic environment inside lysosomes. Now, there are certain conditions that are related to deficiency of any one of these enzymes, okay? And these are collectively known as lysosomal storage diseases, and there are different types depending on the affected molecule. So you have glycolipidosis, meaning that there is a problem in the cleavage or degradation of glycolipids. Oligosaccharidosis, from the name you can tell what this condition is all about. And you have mucopolysaccharidosis. That is basically when you have deficiency in hydrolysis of glycosaminoglycans, like enzymes that degrade heparin, keratan sulfate, dermatan sulfate, and so on. Okay. Now, overall, these are really severe disease. They can be severe diseases, and usually they are associated with mental retardation as well as premature death. One of the most common diseases is Gaucher disease, and there are different types of Gaucher disease, and Gaucher disease is caused by deficiency of the enzyme acid glucocerebrosidase, which is the enzyme that is responsible for degrading a sphingolipid known as glucocerebroside. Okay, so so what happens in individuals having this deficiency is that they are unable to degrade or to grade the glucocerebroside, the glucocerebrosides that are present on the plasma membrane of red blood cells once they are phagocytosed, once they are eaten by macrophages. So they accumulate. There is accumulation of glucocerebrosides. Now there's another condition known as eye cell disease, which is caused by deficiency in the targeting molecules or the molecules that target lysosomal enzymes from the Golgi apparatus. So these lysosomes would be empty of these of lysosomal enzymes. Okay, of course, again, it's associated with retardation. So basically we talked about vesicular fusion with the plasma membrane. Now the opposite can take place, which is also known as endocytosis, the opposite of exocytosis. So endocytosis is when you have vesicles formed from the plasma membrane and they get into the cells. Now what happens is that you have the formation of vesicles. They pinch off the plasma membrane, and these vesicles can fuse with early endosomes. Okay. Now what happens then is that if you have endocytosis of a receptor that is bound to a ligand, the ligand is released in early endosomes and the receptor goes back into the plasma membrane. So there's recycling. Okay, we'll talk about it in few minutes. Now what happens is that these early endosomes can mature into late endosomes, and late endosomes can fuse with vesicles forming lysosomes. These vesicles that contain the acid hydrolases. Okay, and in addition to the pump, the environment becomes very acidic inside these lysosomes. Now, phagocytosis is a type of endocytosis, except that the vesicles that are formed from the plasma membrane are large. Now, phagocytosis means cell eating. So what happens is that you have entry or endocytosis, or eating of a large particle like a whole bacteria, for example, into a vesicle known as phagosome. And these phagosomes can then fuse with lysosomes. Again, you have these acidic vesicles containing all of the proteases and all of the enzymes that degrade bacteria. Okay. In contrast to phagocytosis, you have another process known as penocytosis or macropenocytosis, which means cell drinking. So the vesicles that are formed are really smaller vesicles in comparison to phagosomes. Now there is an interesting process known as autophagy. Autophagy is self-eating. Now this is an interesting phenomenon. The discoverer of autophagy got the Nobel Prize a few years ago. He's from Japan, and the idea is that what cells do if they are under stress is that they can form intracellular phagosomes, and these phagosomes can eat the cell itself. They, like mitochondria, for example, they engulf a whole mitochondria and they degrade these mitochondria. And the reason is that to allow cells to have enough nutrients and the basic molecules, the building blocks like amino acids and small sugars and so on, to build, to generate energy and to build newer molecules. Okay. Now, there is a mechanism that I just told you about, which is basically if you have endocytosis of a receptor with a ligand bound to it. This mechanism is known as receptor-mediated endocytosis. And the idea here is that you have a receptor on the cell surface. what happens is that the ligand binds to the receptor. That results in, first of all, signaling. Okay. Two, you have engulfment. You have the formation of a vesicle with the help of a protein known as clathrin. Okay, we just talked about it a few slides ago. And this clathrin facilitates the formation of a Clathrin-coated vesicle. What happens then is that you have clearing of clathrin from these vesicles. The vesicle fuses with the early endosome. Now the ligand dissociates from the receptor, and the receptor is recycled back into the plasma membrane. The ligand, on the other hand, is transferred from early endosomes. They mature to late endosomes, and then they fuse with they mature into lysosomes, where the ligand is degraded. So this is a mechanism by which cells can control the intensity of the signal. So the signal it is terminated once the receptor with its ligand go into the cell, and also that regulates how many receptors there are on the cell surface at any one time. Now, problems in receptor-mediated endocytosis are associated with a number of diseases. One of the most common ones is known as familial hypercholesterolemia, where basically there is deficiency or inability of the LDL receptors to bind to LDL, or for the LDL receptors, once they bind to LDL, to get into the cell. Okay, and that is caused by an amino acid change in the LDL receptor. Okay, so what happens here is that why is that dangerous? What happens is that the LDL is the carrier of cholesterol. If it doesn't get into cells, cholesterol would accumulate, or LDL would accumulate in the blood, and it has a tendency to cause atherosclerosis. Okay, so you have these clogs of cholesterol that can block blood vessels, and that would cause, again, atherosclerosis and heart attacks and all these diseases.